Matt gave me changed the title. I gave him one title. He gave me this, and I like it better. So, really, it's looking at what we've seen from SNP chips and our expectations based on what we know about SNP chips. When we went from the 50K up to the high density, we thought that having more SNPs on the chip would get us closer to the variants we're really looking for. Allison mentioned causative variants that are what we're really after. Is right now we know that QTL exists. We don't know, for the most part, we don't know what they are and what the other variants that actually cause them are. But going to the high density, we thought we were getting a lot closer. In this particular plot, at very short distances, we have strong correlations between SNPs that are not separated by very, very much, and as that distance increases, that correlation drops quite a bit. So then we've been getting sequence data. This is out of the 1,000 Bull Genomes project, where they reported imputation accuracy from high density to sequence level of averaging around 75%. And most of what we see for imputing from 50K to high density is well over 90%. Generally, there's some groups report 98, some 99, some 97, and they're all up there. And so why is this so much lower? Then what really raised the red flag with me was from some of our sequence data, we see quite different distributions of allele frequency, where the SNPs on the high density chip are generally moderate to high frequency, where all of our sequence variants, most of them have low minor allele frequencies. And we need similar frequencies to have high strong LD or high correlation between the two, and the maximum we can get between the average high density chip and the average sequence variant is about 0.35. So that there's something with the allele frequencies on SNPs that were selected to go on the high density versus what we're actually seeing in sequence. So that's not real encouraging for being able to impute from high density to sequence. So given that, I thought it would be worth taking a closer look at what we actually do see in LD patterns between the high density chip and our sequence data. What I used for this was, we have 176 bulls that, we have high density genotypes and exome sequence that averages about 20x over the protein encoding region of the genome. And this would include 123 bulls that were sires of cycle 7 breeds in the GPE project, and 53 of their sons that were F1 bulls that sired a number of calves in GPE. I limited it to both the high density and minor and sequence variants that had minor allele frequencies in this set of bulls, and looked at, and for the most part I considered non-synonymous SNP to be representative of the set of coding sequence variants that we detected in sequence. And use their actual chip genotypes and the sequence and the genotypes called from sequence, and looked at various pairs, LD between high density SNP on the same chromosome, and then between high density and non-synonymous across the whole genome. Using the full set of bulls as well as 2 sets of purebred bulls, both the Angus and Hereford. Let's see, we see a similar LD decay pattern, between high density SNP, but that's a lot lower between high density and non-synonymous. This is pretty close to that 0.35 we had for the maximum LD we might expect. And that carries out to 2,000. When we get 2 megabases of separation, we're down at a baseline LD that you can still have some correlation between well-separated SNP. But what's really interesting, at least to me, was the distribution. Where this is, these represent means within an interval, but looking at the distribution of LD values within the interval. In this case, we're looking at SNP that are separated by less than 2,500 base pairs. And the high density, most of what we see are imperfect LD, and there are a few that have no LD and not much in between. And it's exactly the opposite between high density and non-synonymous, where we have less than 2,500 base pairs between a high density SNP and a non-synonymous SNP. Most of those have no correlation between genotypes, and there are a few that'll be fairly highly correlated. And the same thing, we see the same thing within both sets of purebreds, the Hereford and Angus. Trying to characterize everything, all the pairs that we did see in high LD, and for that a correlation of 0.9 between genotypes would correspond to an R-squared of 0.81. 75% of high density SNPs have high LD with some other high density SNP. Several of them, about a third of them are highly correlated with several high density. 80% of the non-synonymous are not highly correlated with any high density. And then a few are highly correlated with several high density. And then among non-synonymous SNP, about a third are correlated with some other non-synonymous SNP, and 2/3 not correlated with any. And looking at separation between those pairs, where we do find high R-squared between pairs of high density or between high density and non-synonymous, most of those are separated by 10 kilobases out to a megabase. And there's not much that, that are separated by less than, definitely less than 1,000 bases or so. The non-synonymous SNP that are highly correlated tend to be a little bit closer together. And just one thing to look at, another thing was looking at overlap among these high density pairs where we, looking at them at all bulls, Hereford and Angus, there's essentially no overlap between what the pairs that are in high density in Angus and Hereford or in the set of all bulls. So, this goes a long way to explaining why our genomic predictions don't work well across breeds. That, if we're depending on LD between SNP and unknown QTL, we're not going to, and we have completely different LD in different breeds, we're not going to be able to predict from one breed to another. But then, besides, our predictions aren't going to be just based on high LD between SNP and QTL, but those SNP effects can be influenced by moderate correlations, and I set these levels to correspond to what we'd consider moderate to high genetic correlations that would change an index value. So between, that would correspond to correlations of 0.5 to 0.9. And within high density, almost all the high density SNP are at least moderately correlated with one other high density, and most with, up to 1,300 other high density across all bulls. Over 3/4 of the non-synonymous are moderately correlated with at least one high density, and half of them are moderately correlated with several high density SNP. And we see still have a lot of non-synonymous SNP that are correlated with other non-synonymous. So there's a lot of low to moderate LD that it could be that is influencing our genomic predictions. So the separation between these moderate LD pairs is pretty similar to what we saw with high LD, where but it probably extends further over, and it could extend from one end of the chromosome to the other, or actually, if we go back here, there's a lot of this moderate LD where we have moderate correlations between a SNP on one chromosome and something on another chromosome. And one thing we've been playing with for a long time was, where we might not capture the QTL signal with a single SNP, but looking at flanking SNP, and so SNP on either side of what we think might be a QTL that might give us a higher correlation than either one of the single SNP. And so I looked at LD between pairs of high density flanking a non-synonymous, either using a genotype model that, just regressing on both the genotypes for both flanking SNP, or, based on our imputation, looking at the two SNP haplotypes to try and predict that, get a correlation between the pair of SNPs and the non-synonymous in the middle. And we get, from either approach, we get pretty much the same answer. There's no cases where the regression on the individual SNP gives us a higher R² than using the 2 SNP haplotype, but we still aren't getting the correlations we'd like to see. The distribution isn't that much different, with low R² between the non-synonymous SNP genotype and what we'd predict from the flanking SNP. So if what we're seeing between our non-synonymous SNP and QTL is typical of what we'd expect for LD between QTL and our high density. There's not going to be that many QTL that have strong correlations with anything we're genotyping with the high density. But most of our QTL are going to have moderate correlations with lots of different SNP. So for genomic selection, that probably isn't going to make a whole lot of difference as long as those correlations are consistent between training and prediction. But it could be misleading on, what we get for genotype and associations between SNP genotypes and phenotype. What we see is a SNP that's strongly associated with phenotype could be the result from that SNP being correlated with, the combination of correlations with lots of different QTL. It could be a correlation with a QTL that's well separated from that SNP. And, are we missing QTL signals that aren't from QTL that aren't strongly correlated with anything that's on the high density or other SNP chip? And if that's the case, are there ways we can avoid the dependence we have right now on LD between SNP and QTL? Can we use the sequence variants to predict, in our genomic predictions? And will that be more accurate than what we get from the SNP chips? Some of the more of the 1000 bull project work, they do get a slight increase in accuracy from imputing to sequence variants over what they get from the HD chip. But that's not going to eliminate all the LD. Using these sequence variants for genomic prediction, they're still correlated with each other, so any effects we estimate will be influenced by correlated variants. But will they be more portable than what we get right now? You don't know. Are there other, what I've seen from imputation results, still doesn't look real encouraging for imputing low frequency sequence variants. So, something like the functional assay that Missouri's developed, or other assays that might give us, better genotypes for these functional variants might be more useful. So, that's what I have.
Questions for Warren? Warren, I've got one. So, and I had to step out for a second, so maybe you covered it. But I think one question might be that people in the audience might wonder about is we're beginning to talk about what we perceive advantages to be from selecting variants from sequence information and putting on custom chips to maybe increase the accuracy of prediction within breed and allow us to predict across breeds. So we've been in this arena of genomic selection since roughly 2009 or 2010. So why now? Why not have taken this approach earlier?
Jerry might answer this better than I can, but I mean, it's the cost of sequencing is so much cheaper now, and when we developed the first chips, we had a list of what we detected and wanted to find something that would be informative. And a lot of populations, so I think frequency was part of the criteria for both 50K and HD design, that where you didn't want to select 50K that would only be that would mostly be informative in Holstein and not across other breeds.